now we're going to go ahead, and we've talked about enzymes, right? And who can tell me what they know about enzymes right now? All right, very good. One at a time. So we know enzymes can be made of protein. Give me a second. And it's most of the time. But what else can they be made of?
RNA.
That's right. Remember, answering and asking questions gets you your 100 points for the week and for the day. Ribosomes are the example there. What else do you know about someone else? Proble?
They change with pH and temperature.
Change what?
The shape, more functions.
High temperature, right? The classic drawing of the graph for both pH and temperature is something like this, where you have an ideal range, what's called an optimal range, right? So this is the optimal range, and this is where, oops. I will zoom in back in just a second. Really, often what happens is that you get a drop in enzyme function. It looks like this. It's almost a falling off a cliff deal. But this optimal range is usually the temperature. Usually the temperature in which come on. Enzyme is found naturally. So if it's found in a beast, in a human being, you'd expect this temperature range to be what? This optimal range. Optimal, by the way, means best. Like 96.5 degrees Fahrenheit, something like that, 97. So anywhere from like 96 degrees Fahrenheit to, I don't know, 98, 99. Let's say 99, you'd probably get okay function. But if you get to way out here, 105, whatever, it denatures. That's right. Anything beyond that, it denatures. So that's going to be key. That's a good question. That's a good question, yeah. It kind of does. But here below, it's not so much the denaturing. Here, there is not enough kinetic energy, right? The temperature is too cold. Remember, temperature is an measurement of average kinetic energy. So below the optimal range, there's just the molecules are not moving around enough. They're not getting to the enzyme. They're not hitting each other. The reaction is not happening as well as quickly. And this is reaction rate, by the way. So that's rate of reaction. I'm just a sketch. So it's not. Don't count this as like how to do a graph. So you see the similar thing with pH. With pH, you see the graph is a little more, Again, with unfortunately or fortunately or unfortunately, here with pH you're actually dealing with denaturing on both ends. If it's not the appropriate pH, if that it's not the optimal range, if it's not the optimal range, by the way, this is optimal, right? If it's not the optimal or max range, then there's denaturing on both ends. Because if the pH is too low or if it's too high, then the hydrogen bonds are broken and the protein unravels. So here there's denaturing on both sides with pH. Give me a second. Now that's important to understand for this part here. Here, the temperature which the enzyme. But here's what's fun about this, or what's good for you, is if it's pH or if it's temperature, if it's outside the optimal range, it doesn't function. It won't work. And with temperature, it's because the protein unravels in high temperature, it misshapes. In pH, on both high and low, they unravel, but irrelevant, it doesn't work. Doesn't work outside its optimal range. So how would you do that? How would you run, if you want to catalyze a reaction, but you want to do it at a higher temperature, what would you do? Well, let's forget about hydrogen peroxide for a second. Let's say we have a reaction like, there's a reason I'm asking you this later. There's something called PCR. I don't know if you've heard of it before, but it's called polymerase chain reaction. We use this technique to make copies of DNA. You've seen in CSI shows where they swab for samples and they test your DNA and they find the criminal that way. Mm-hmm. Well, they're using this stuff called PCR. If they find one single hair, they can make millions of copies of your DNA from that one single hair, and they can sequence it. And we use something called PCR, which heats it up very, very hot so that the DNA unravels, right? Because we can denature DNA too. We can unravel DNA. That opens up the code for us to read. But now you need an enzyme that works at that temperature. What do we call, what do you think, where might we get that enzyme? An enzyme that works at like 105 or 106. How can we do that?
Maybe like
degrees. Yeah. Anybody? Fahrenheit, rather. Say what?
Like where can you find that?
Yeah, we can raise the temperature, but where would we get an enzyme? Could we get it from us? Can we get it from our body?
You can get it from an animal that has.
No, because we don't have an animal or some kind of beast. Yeah. You have to get an enzyme that works in extreme temperatures, so you'd have to find an animal that lives in extreme temperatures. That's a really good idea. But actually, lizards that live in deserts and such, they don't do well in heat. They cool off, like going in the shade. They wait until the right time, and they do everything in between the hot and cold, and that's how they monitor their temperature. They still have a more limited temperature. You might look in a volcano, but you probably won't find many living things in a volcano.
Things that live around it.
Yeah. Hey, hello. Love your shirt. Yeah. An animal, but what animal? Huh? A camel's a good idea. But see, the thing about mammals regulate their temperature. So when they get hot, they cool off by panting. They cool off by sweating. Like you still keep, even people that live in the desert, their temperatures aren't higher, right? They just sweat more, and they have clothes. They have something called. Have you ever seen the Bedouin?
No.
They wear like black or white. They're wrapped up from head to toe, and they're in the desert. You ever wonder why they're wrapped up in the desert?
Doesn't black make you sweat more?
Huh? It does protect their skin from the sun, that's true, but there's something else it does. it circulates the air, cools them off. They have their built-in air conditioner. So as they sweat, the air circulates and cools them off. So they've designed that over time. People that wore that clothes survived. Those that didn't. So, but the point is that it cools them off. So it's actually cooling them even though it's wrapping them up. So, yeah.
Maybe a creature from that lives near a thermal vent.
That's right. That's exactly it. You got it. A thermal vent or any kind of, we call these creatures thermophiles. These creatures live in very hot, almost boiling water. They were first found by a biologist in a thermal vent in a national park in the West.
Oh.
They're bacteria. These particular ones are bacteria. You can go to ocean, thermal vents in the ocean and find others.
They were found near the geysers.
Near the geysers, exactly right. So he went to the hot pools near the geysers, and he took samples, and he found bacteria, and those enzymes work at high temperatures. So we've isolated those enzymes, and we're able to...
Thank you, sir.
You're welcome, thank you. So we're able to use those enzymes to our advantage. We can unravel proteins and nucleic acids that are in us that unravel at 102, 105 degrees, but then we can use their proteins to copy them that are not unraveled at that temperature, or denatured at that temperature.
The geysers at the national park, are they being cut? Like, how do
They have all right, that's a little bit off topic, but to be clear, the geysers in our national parks, what happens is that there's magma near the surface, and there's pools of water sink down. Water goes down into the holes. It rains, water goes into those holes, and it fills up, and the water heats up, starts to boil. Pressure keeps building until finally, whoosh, up comes the water, and then it settles back down like a tea kettle. You get it hot enough. I said all those wonderful stories and didn't record. Oh well. Did we get that? Is everybody?
You go back over.
Where?
Yeah, right here.
Wait, here or here?
over there? Yeah.
The-
You like my voice? No. Stop. It's on. That's like optional at the end. Optional. That's what they say, right? So the two months.
Okay,
So that's it. That's good. Well, that's the stuff we know about enzymes. Of course, there's a few other things like what? What is something else you might that's important? They speed up reactions. That's really the important part. They speed up reactions.
They can be reused.
That's right.
They could use-
They don't run out.
They have active sites.
They have active sites. You guys know a lot about enzymes.
That's
They reuse active sites. They speed up reactions. They're not used in the reaction. I heard someone say. What is used in a reaction? The substrate. That's right. Substrate's used. And we know one more thing. Enzyme is relatively specific for the substrate. Substrate's the thing that it acts on, right? The reactant that it helps speed up the reaction for. They match very well. And that's pretty good. That's a pretty good summary of everything we've done so far of enzymes. You guys did a great job. Yeah.
So does that mean there's different types of enzymes?
Say what now?
Does that mean there's different types of enzymes?
Absolutely, there's thousands of different enzymes.
Like different shapes.
Doing that, different shapes. Each one matches a different molecule. If you have a molecule, you have an enzyme that matches that molecule. If it's going to be acting in your body. Now, to be fair, some enzymes work on a family of molecules, right? So not all enzymes are specific to a single molecule, and some there are. I did this in the video, so I'll let you watch the video. But there are. We do use chemicals to mimic the substrates. Like we know what the shape of the substrate is, so we design a molecule that fits into that enzyme. And we can do that to design weapons like poisons. Neurotoxins they don't work on an enzyme, but we're able to kill you by making a mimic of a molecule that would normally fit into a protein and send a signal down your neurons. Well, if we mimic that and we stop that and we lock up your receptor, we can stop your neural pulses, so that freezes your muscles, your heart stops, your muscles twitch. We call that a neurotoxin, or what is it? I forget, whatever the neural weapon is called. I forget the actual name for it, but it's a toxin that we designed to impact your nervous system. I say we, I mean humanity, not me personally. But these weapons are every major nation has said they won't use them, but they all have them. So they're really scary stuff. They'll kill you in seconds. They'll stop your heart as soon as it touches your blood. Just by touching, a single drop touches you, it's over. So it's an interesting, horrible invention. All right, horrible use of this lock and key method, right? All right.
Why was it invented?
God, I ask myself that question all the time. People go to war. People are always paranoid. They want ways of killing their enemies and defending themselves. And so they design weapons to do that. Why we do it? Why can't we, as Rodney King once said, why can't we just all get along? Why can't we just everybody stay inside their border, share whatever we got to share, and live our lives in peace, and maybe even try to expand humanity beyond this planet? Actually have a planetary goal of finding life on other planets, moving faster than the speed of light, right? Wouldn't that be nice if we just all work towards those goals instead of just worrying about this nonsense, the stuff, the ridiculous things we all worry about? If we all just let each other live, right, and just mind our own business, and try to there's all these wonderful ideas that often come from children, right? The problem is our system, the way it's designed, sets up competition for resources, right? And all life, as we'll learn in ecology, is competing for resources. All life wants to live even if it means others have to die. And that's why we eat cows and chickens, and we might feel bad, but we're going to continue to do it because we live, and if they have to die, that's it. So unfortunately, that's expanded and intervened in humanity as well. And we'll use all kinds of things to justify it in our history, in our society, right? It could be nationalism, it could be religious fervor, it could be racism, it could be, 1,000 things. But nobody ever remembers the one thing, like, why do we have to kill each other, right? Like, why do we have to do that? I don't know. I don't know the actual answer for that. It's a good question, though. All right. Now that I'm depressed, let's go on and talk about the next. What is the? So we know how enzymes work, and we know that. Let's look at. We're going to go on and look at how. Now that we have an idea of how enzymes work, we can look at a specific chemical reaction. And so let's look at photosynthesis. And tomorrow you'll get a podule on it. Now, photosynthesis happens in eukaryotes. That's right. And mostly in eukaryotes called plants. But not just in plants, though. There are also prokaryotes.
Bacteria?
Yeah, bacteria also do photosynthesis. And there are some protists. Let me double. There are some protists. Protists are eukaryotes. They have a nuclei, but they're not plants. They're single cell. Remember, we talked about the different cell types. Please, a lot of you have had that assignment excuse, but please get that assignment done on cell organelles. It'll help. It'll solve so many of your issues. Thank you. Protease. So, protease. Protists. Tists. Correct. So, in a plant is made of has a cell wall and has a membrane. It has a nucleus, a nucleolus, endoplasmic reticulum, rough and smooth, the Golgi body, vacuoles, mitochondria, has another thing called chloroplast, and I'll do that in green, and you'll see why in a minute. And a protease has very similar things, except the thing about a plant is that a plant is multicellular. They sit next to each other, the cells grow, they multiply, they have they form shapes, etc. Right? There's millions of cells in a plant. A protease, so a plant, a protease is usually single cell, and they look. They have different shapes. They have different. Some protease might even have a flagella. Some might have cilia. All right. Flagella, those little tails that help them swim, and the cilia are little extensions of the membrane that help them move along. But they have inside them they also have a cell wall, but their cell wall is not made of cellulose. Many of them have cell walls, if not most of them. They have something called a contractile vacuole, where plants have a central vacuole.
This is for the prokaryote.
The protease. Protease. Prokaryotes don't see. That's the danger. I'm gonna tell you right now. That's the danger. Prokaryotes are basically bacteria and archaebacteria. They have no nucleus, no internal membranes. They only have the cell membrane. That's it. Give me a second. No contractile vacuole, no nucleus, no chloroplast, no
What do they have?
mitochondria. They have DNA. They have ribosomes. They have other molecules, but they don't have all the organelles that eukaryotes do. Does that make sense?
So basically, it's like the eukaryote is kind of more like related to like a human cell. 'Cause
Yes,
it's an animal,
that's right. That's right.
where a prokaryote is just like
A bacteria and archaebacteria. That's right.
a plant and grass and stuff.
Plants and grass are eukaryotes. Yeah, they have nuclei. And bacteria are the only ones that don't have
I
it.
get it now.
Yeah.
Why do prokaryotes have DNA but not nucleus? Isn't not nucleus? A mitochondria. Isn't mitochondria when you say nucleus?
Yeah. Let's talk about that a little bit. First, let's talk about this. So protease have also a contractile vacuole. They have a nucleus, nucleolus, endoplasmic reticulum, mitochondria. Some of them have chloroplast. Some of them do photosynthesis. Others don't. All right. So protease are a really broad family. They're all eukaryote. Both these groups are eukaryotes. They all have nuclei. Yes. Guys, the only ones that don't are prokaryotes. That's it. The only ones that don't have a nucleus, don't have organelles, are bacteria and archaebacteria. They're both basically bacteria. Archaebacteria. Meaning they're older bacteria. These are the bacteria we find in extreme conditions, high pH, high temperatures, really weird places. that makes sense when you think of it in a way in context of evolution. If life started on planet Earth the way we think it did through using evolution, then you would expect that then, if that's true, then you would expect that the most extreme life must have existed in the most extreme circumstances. The earliest creatures must be able to do that, and they are in fact archaebacteria. Yeah.
So are archaebacteria found in the glaciers or the frozen?
Yeah, the glaciers, the bottom of the glaciers. These guys are found in hydrothermal vents, in saline, high salt concentrations, high pH concentrations, low pH concentrations. They're found in the continental plates. I mean, they're crazy places you find them. But anyways, they're all prokaryotes, which means there's no nuclei. But they can still do photosynthesis. They just do it across their membrane. They use their actual membrane to do photosynthesis. They don't have chloroplasts. They do not have chloroplasts. Is that clear? They do photosynthesis within their cell. They have a DNA. The DNA is round. It's circular. It's called a plasmid. They have smaller versions of the DNA that are also round. This is DNA. These are not organelles. This is DNA. They have DNA. They have RNA. They have ribosomes. Ribosomes don't have membranes, so they have ribosomes. And they have all the components to do the stuff that mitochondria do. They do all this. They can do. Some of them can do all the stuff that not all bacteria can do photosynthesis. Some can do the stuff that all of them can do the stuff that mitochondria do. They can do cellular respiration. Some bacteria can do. I'm gonna do this one in green. Some bacteria can do photosynthesis. They do not have chloroplasts. They have the circular DNA. They have the smaller circular DNA. They have ribosomes, and then they have the chemicals necessary to do photosynthesis and enzymes necessary to do photosynthesis. And this one has and they can also do cellular respiration, by the way. They can do both.
Prokaryotes?
These types of prokaryotes, yes. So some prokaryotes can do cellular respiration, like mitochondria. They don't have mitochondria, but like mitochondria. And some prokaryotes can do photosynthesis, even though they don't have chloroplasts. Or mitochondria, and they can do both photosynthesis and cellular respiration. Other prokaryotes can only do something called chemosynthesis. They can't do cellular respiration. They also have a DNA, small and large. They have ribosomes and all the chemicals necessary to chemosynthesis, the way of making molecules using just chemistry. They don't use oxygen. They don't use sugar. They don't use the light from the sun. They can use chemicals to make ATP, chemicals they find in their environment. Yeah. Yeah.
I have a question. The word chemo, is it close to how they use chemotherapy?
Yes, in both chemotherapy and chemosynthesis, both of them are using chemicals. All right. One is to create chemosynthesis. The other is chemotherapy is to kill cancer, hopefully. Unfortunately, you also kill your cells, but that's another story.
Is that how you lose your hair?
Yes. That's one way. Radiation therapy also does that. All right, so in eukaryotes, when it's talking about photosynthesis, we're talking about an organelle that's called, we're going to talk about an organelle that's called a chloroplast. Chloroplasts have two membranes. Not one like the rest of us do, like the other eukaryotes do. They stack their membranes, the internal membranes, they stack them in these things called thylakoids. There's a lot of them. They pack them. Their whole job is to make sugar. A single stack. They're like pancakes. Think of them like a stack of pancakes. You've all eaten pancakes or seen them, correct? Whatever. They stack of pancakes, stack one on top of the other. Each pancake is called a thylakoid. And a thylakoid, a whole stack of them, a stack of thylakoids, we call it, hold on, give me a second, a granum. A granum. So a single thylakoid or a stack is called a granum. Chloroplasts are found in plants and protists. Plants and protists, right? They have these little green, they're all green. What can't you see? Granum. Granum. Granum.
Chloroplasts go under eukaryotes? Yes. I was asking to make sure.
Yes, chloroplasts are only found in eukaryotes because, guys, prokaryotes don't have any internal organelles except ribosomes. They don't have a nucleus, hence prokaryote. They don't have mitochondria. They don't have Golgi body. They don't have endoplasmic reticulum. They don't have vesicles. They don't have any internal membrane organelles. They have DNA, they have RNA, they have ribosomes. That's all, folks. And chemicals. They're very basic creatures. They can be very complicated, do all kinds of amazing things, but they're not as complicated as a eukaryote. Is that clear? All right. So chloroplasts have two membranes, thylakoid and granum. The granum is a stack of these thylakoids, and the two membranes they help exchange, turn light and carbon dioxide into sugar. There's two sets of reactions in photosynthesis. So in photosynthesis, there's two sets of reactions. There's the light dependent reactions. And it used to be called the dark reactions, but we no longer call them that because it confused people. Thank God they changed it. They don't happen in the dark. They can happen in the dark, but they don't necessarily have to happen in the dark. They're called the light independent reactions. Light dependent and light independent reactions. It can happen in the dark, often happens in the dark, but there are some plants that have evolved to work where they do the light independent reactions at a different time because of the heat. And we'll talk about that when we talk about the different kinds of photosynthesis. There's three kinds of photosynthesis. You should have some idea how they work. I honestly don't think they'll test you on it on the OST, so I'm not too worried about it if you get them, but I think you should be exposed to them. And the coffee's good. All right. So the light dependent reactions. What light do you think is used by photosynthesis?
The sun?
Yes, it's going to be sunlight. And so let's talk about sunlight. And we learned about pigments. There's a reason we learned about pigments. I did it on purpose. I did it on purpose. All right, so sunlight. What colors are in sunlight?
Yellow. White.
Are we just going to guess? White is not a color. It's a combination of all colors. It is, sunlight is white. White means, white light is all the colors put together.
I thought it was yellow.
What are the colors?
Rainbow.
So write this down. It's an easy way to remember. Roy G. Biv. Roy G. Biv. Red,
Rainbow.
orange, yellow, green, blue, indigo, violet. The colors of the rainbow. When the colors that you see when light goes through a prism, that is the color, those are the colors in white. When you see white light goes in, it splits light, a prism splits light into all the colors, red, orange, yellow, green, blue, indigo, and violet. Those are the colors that you have to deal with. It's this visible light that that photosynthesis takes advantage of, all except which one? Black is the absence of light. Black means that all light is either there's no light there or all the light's being absorbed. Okay? So when you see black, that's the absence of light. White is all the light. Black is the absence of light. So it's, even though it is a color, when we talk about color as a normal human, as a scientist, black is not really a color, it's a lack of color. Because light is color to us in science is a frequency of light. All right? Green. That's right. How do you know it's not green? How do you know that green is not being used?
Because the plant's green.
You know right now, if you think about it, that green is not being used by plants. How do you know that?
The plant's green.
Because the plant is green. What does that mean?
It has pigments. It took away the green pigment.
It didn't absorb the green pigment. Because when your eye looks at it,
I see green.
you see green. Green is the light that comes back to your eye. White light goes in, sunlight goes in white. All these colors go in. Red, orange, yellow, green, blue, indigo, violet all go in. This plant absorbs all the light except for green, and that's the color that you see. If it did absorb all the light, what color would plants be?
Rainbow.
Black.
Black.
If the plants did absorb all light, then they would be black, but they're not. They're green, and that's the color you see. Yeah. What's happening in the fall?
It's orange.
Like a sunlight.
No, yes, like a sunlight. And so what happens to the leaves?
They don't.
Why are they changing colors?
They absorb the green pigment and don't.
Why do we call it fall?
Because the leaves are falling.
Because the leaves fall.
They die.
And they
They're
change
dead.
color.
Yeah, they're dying. They're dying, and the chloroplasts start to break down. The various pigments start to break down, and as you start to break down, the ones that last the longest are the yellow and the red pigments that absorb everything but yellow and red. Those last the longest, but the others, the ones that absorb indigo and violet and blue, they tend to die. So you don't see, they tend to unravel. They tend to be broken up faster. So what you're left is with the ones that absorb everything but yellow and orange and red. So this half of the rainbow, that's why you don't see like purple leaves falling, or violet or indigo or blue leaves falling.
You've ever seen purple lettuce?
Oh my goodness. Can you be quiet?
No, it's that.
All right, so she's been distracted since this morning, since she ran into a painting on purpose. So anyways,
Wait, no, y'all never seen like.
Carl, yeah, it's purple lettuce. Is lettuce? Yes, there is purple lettuce. Yes,
Did it absorb the violet?
it's a long story. Let's not get into the exceptions. Let's figure out the rule first, and then look at the.
Sunlight go under.
What?
It's supposed to go under light, depending or.
I feel like I should get these kids some cocoa pebbles and sit them inside. Could y'all pay attention, please? Shh. Shh. So there are purple plants, right? So that's another story. We'll talk about that. That's an exception. In general, green plants are absorbing everything but green. They're more common, and let's stick with the rule. So light is going to be absorbed, and that light, along with water, that light is going to be used in what we call photosystem. Photosystem 2 first, yes, and then it'll be used, and then again in photosystem 1. It's annoying that that photosystem 2 happens before photosystem 1. I don't know why. Maybe they discovered photosystem 2 after photosystem 1. I don't know. For years I've said, why don't they just switch the names? Because people learned them this way and they don't want to switch it. All right. So sunlight's going to come in, and you have inside the chloroplast, you have a membrane. As we discussed, I'm going to go ahead and draw a membrane. You know what?